Just
So
got it.
I'm sorry.
Okay.
Hi, Paul.
Hi.
Sergey Piligin.
Hi.
I just want, you were talking about the data-free modeling, and I just recall that some years ago, I was a postdoc at Emory, and somebody said a very, very interesting observation that data is the crutch for an insecure modeler.
Well, I'm glad you said that and not me. I think there's some element of truth to that. I think it's gone too far, and I say it partly driven by funding agencies, but it's also the problem with biology is that the data is often not very reliable, and you can overfit to the data. So of course, I'm not saying one should ignore the data, but I think there's a lot of scope for looking at the cartoons at the end of experimental papers, which summarize what the experimentalist thinks going on under the hood. And I think that's a nice place to start modeling. And so when I say data-free modeling, I mean starting from the cartoons and developing a conceptual toy model, which allows you to try and test hypotheses and to develop and identify mathematical and biophysical principles that don't only apply to one system, but be carried over to multiple systems. And so it's more qualitative rather than quantitative modeling. So that's where I'm at. So when I give a talk and then somebody asks me, have you tried to fit your model to data? I'll tell you now, no. So that's not what I do. Hopefully people will be interested in some of the work I do and do that. And I'll mention some work in my talk, but that's not what I'm about. Okay, so let's now go to some of these conceptual frameworks and cartoons. And so I've been interested in a while now in morphogenesis, and the classical picture that comes from a fellow Brit, Lewis Wolpert, is the French flag model, which basically says that, and now as an applied mathematician, I treat an organism as just a one-dimensional line, which is already moving away from the data, but we've got a body axis, and one imagines there's a source of messenger RNA at one end of the axis that synthesizes proteins that then diffuse in the extracellular space, and as they diffuse, they attach to receptors on the surface of cells, which can model as a degradation. And so in steady state, you get a concentration gradient. And the idea is that this concentration gradient is thresholded so that depending on the value, one sort of gene or another is switched on or off, and that leads to cell differentiation that's correlated with body position. And this has been studied greatly, both experimentally and mathematically. From a modeling perspective, one's interested in how robust things are, the time it takes, because if it takes the lifetime of the universe to form a concentration gradient, it's not going to be a good model of the system. And that's where qualitative modeling comes in, because you at least want things to be consistent with length and time scales of the systems you're trying to model. But what I've been fascinated in for quite a while is the following observation that has been driven by an experimentalist, Kornberg and collaborators. And it's the observation that many cells in developmental biology extend actin-rich filaments called cytonemes that are very active, growing and shrinking, that make direct contacts with other cells. And although they're very hard to image, they can extend up to 200 microns. They have diameters of, I guess, 10 to 50 nanometers. So it's quite hard to image, but you can actually see myosin motors and other things moving along these fibers, transporting cargo. So the hypothesis is that these guys, through direct contact, transport morphogens or the cognate receptors and facilitate morphogenesis. And there was virtually no modeling work on this topic. And it's something that I think there should be more done on this, because it's becoming more and more prevalent in experimental studies of embryogenesis. And the other thing, and so if one now wants to start writing grants, and I still haven't tried this, but it's becoming clear that related filaments also seem to play a major role in direct contact of viral spread, including COVID-19, where the virions promote the growth of filaments, and then virions use these filaments to infect other cells. And if I have time at the end, I'll briefly talk about the beginnings of work I'm doing with a master's student on developing that model. I should also say, and this was just a cool poster that I found recently, so a lot of us are used to the notion of microtubular filaments, playing a role in cell mitosis and many other things. But there's now a growing number of systems where actin-rich filaments are playing a role. And they're often called tunneling nanotubes or and so forth. But in the context of embryogenesis, they're called actin cytonemes. But they often play a similar role or behave in a similar way in spite of or irrespective of the system. So it seems to be a very interesting process where now the notion of growing and shrinking actin-rich filaments rather than tubulin-rich filaments is something I think worth pursuing. Okay, so carrying on with the cartoon theme, another interesting thing that's emerged is that the mechanism for transporting morphogen or the corresponding receptors seems to differ depending on the organism. In particular, in invertebrates, such as Drosophila, it seems to be that the distribution of morphogen seems to be mediated by filaments that form quite long-term direct contacts and transports via myosin motors. But even there seem to be two different types of contact. You can have a very explicit direct contact, like a tunneling nanotube, but there's also growing evidence that some of these contacts look more like synapses, where you ultimately have a release of a vesicle of morphogen that attaches to the receiving cell. The other mechanism that seems to be more prevalent in vertebrates, such as zebrafish, is more what I call a search and capture process, where the filaments are much more active and grow and shrink as if they're searching a space looking for a target. When they attach to a target, they only stay attached for a little while, and the morphogen tends to be located on the tip of the filament and is then released into the target cell. And so this is quite analogous, actually, to microtubules searching for kinetochores during cell mitosis. And the mathematics is actually quite similar. So that's the kind of basic framework that I want to talk about on the modeling side. So what I'm first going to do is briefly talk about a direct contact model, which I looked at in the past, but most of the talk's going to be focusing on the search and capture model, partly for reasons that it has more interesting mathematics, but also I think is a richer system. But let's just briefly talk about the direct contact model. So this idea is that the cytonemes have contacted target cells, and for simplicity, we'll imagine an array of target cells, and the source cell is sending morphogen, producing morphogen at a rate Q, and then these guys are being transported to the target cells and being received. Already, excuse me, thinking about the cartoon makes one start ask biological questions, because the minute one has a setup like this, one can start asking, well, there looks like there could be an additional control mechanism, and one can speculate that one can control the morphogen gradients at a cell signaling level by sorting out and assigning relative rates to the different cytonemes in the source cell. And so therefore there could be a higher probability of sending morphogen to one cytoneme rather than another. That hasn't been tested experimentally, but it is something that allows an extra freedom of control in this sort of setup. The mathematics is pretty simple. It's a compartmental model. And at the compartmental model level, let C naught be the concentration of morphogen in the source cell, and it's produced at a rate Q, and then it's lost through the fluxes entering the various cytonemes that are then transported to the target cells. And so the concentration in a given target will be the flux arriving to that target along a cytoneme of length Ln. And then there's, of course, utilization or degradation of the morphogens that's proportional to the concentration. So that's a compartmental model, which is simple to solve. But the next level of modeling is to say, well, how do I determine these fluxes? And this is where one starts modeling motor-driven transport. So there are different levels that fit together for the full picture. And so the classical transport equations are basically these advection equations. And these come up a lot. And they also used to describe the growth and shrinkage of microtubules in catastrophes and so forth. So these are velocity jump processes. And the idea is that you have two populations. You have a population of motors along the nth cytoneme moving towards the target cell, anterograde transport. And then you have another population that's moving back to the source cell, retrograde transport. And It's known experimentally that one has complicated motor cargo complexes that involve a mixture of forward-moving and leftward-moving motors that could be on opposite polarized filaments, for example. And so there's a switching between the two populations. So in terms of these equations, you have an advection term in the plus or minus direction, and then you have switching rates between the two populations. The flux is then going to be the velocity times the concentration in the plus direction minus the velocity times the concentration in the minus direction. One then has boundary conditions saying there's a rate of injection of flux from the source cell, and we treat the target cells as absorbers. This is now a complete dynamical system, and one can then solve them. This is something I hope one could still give an undergraduate to solve. So one can work out the steady state equations and just plot the solutions. And lo and behold, as one might expect, just as in diffusion-based morphogenesis, you get morphogen gradients in the steady state. And their behavior depends on whether the net transport is in the positive or minus directions. This is a bit counterintuitive, and I'm not claiming this is the correct way things work. But what we discovered when we looked at this is we didn't just look at the stationary distributions, but we started asking questions like, well, how long does it take to set up the morphogen gradient? Is it consistent with biological timescales? And more importantly, how robust are these things? And what we discovered, so we looked at robustness in terms of, suppose I perturb the rate at which morphogen are produced in the source cell, how robust is the positional information of the morphogen gradient? And what we discovered was that if one had a dominant retrograde transport, that was more robust. So it looks like, at least within the context of this simplified model, there's a sort of competition between having greater robustness and being less energy efficient by having dominant retrograde transport. So that was a kind of prediction of the model. It hasn't been tested, but this is the sort of thing that came out of this simplified conceptual model. Now, one interesting thing that, Hyung-Jung Kim was my graduate student who did this work with me. And then he did a postdoc at Penn State, not Penn State, UPenn, working with Maury and Plotkin. And they did a nice piece of work where they asked something more, a biological question about, well, what are the advantages and disadvantages from a cost effectiveness perspective of diffusion versus cytoneme-based transport? Now, they looked at a simplified process, but they basically asked the following. They wrote down a total cost function. I won't give the details, but just say it in words that involved the time it took to achieve the concentration gradient, that cost a certain penalty, and the amount of energy needed to achieve the concentration gradient. So in the case of a cytoneme, they broke it up into two phases: a search phase, where a cytoneme is growing and shrinking, and that costs energy through polymerization, and then a transport phase, which again costs energy in producing and transporting the morphogen to a target cell. In the case of diffusion, again, there's the cost of the time it takes for the diffusive molecules to reach the target, and there's also the cost of synthesizing the diffusion molecules. And of course, because diffusion is much more wasteful in a sense, you have to produce more molecules than needed in order to achieve the successful concentration gradient. So they fed all these features into their optimization model, as I summarize on this page, and then compared the two cases. And I'll just summarize the results. What they seem to find is that direct transport, in terms of the cost functions they define, of course, it doesn't mean these are the correct cost functions, but within their work, they found that direct transport is more efficient when n is large. And they also calculated the critical value of the number of morphogen needed to set up the concentration. And they asked, well, what's the critical value where both mechanisms are about the same? And they said, and they characterized the efficiency and this critical number as a function of the distance of separation between the source and the target cell. So I think there's a lot of interesting stuff to do on this side, because there are clearly two very distinct mechanisms of setting up morphogenesis and cell signaling: diffusion-based and direct contact-based. And these are just preliminary studies suggesting there's a lot of interesting things still to understand about the relative efficiencies of the two and why sometimes one is better than the other. So that's all I'll say about that. What I want to do for the rest of the talk is talk about some of the mathematics that applies to directed search and capture processes. Now, these sorts of models I've looked at in a lot of different contexts, also axonal transport, motor cargo based, and also microtubule based cell mitosis, where you have microtubules searching for kinetochores to start pulling apart chromosomal pairs. But I'm going to talk about this. The mathematics is the same, but I'm going to talk about this within the context of the cytonemes. It's funny. So although I say I keep a distance from data and experiments, I'm actually married to a neuroscientist who's a very good experimentalist, Alessandra. She works on primate vision, and she's always nagging me to do modeling and work with her. And I kind of balk at the idea, although we have collaborated. And she can never understand how I get excited when I use the same mathematics and apply it to very different things. But anyway, so what I like about mathematics is it can be ported to very different applications because the underlying conceptual structures are the same. Okay, so let's look at search and capture within the context of cytoneme. And I should say, I'm going to simplify things by saying there's a single source cell and there's only a single cytoneme growing and shrinking from this source cell. One can, of course, make this much more realistic by having multiple source cells and multiple cytonemes, which I'll talk a bit about later. But let's try and get things going by talking about a single cytoneme. So this cytoneme nucleates from the source cell and starts growing in a particular direction. Let's make it more realistic and just not talk about one dimension, but imagine a two-dimensional or three-dimensional tissue with the distribution of target cells. Clearly, each time the cytoneme grows, and of course, there'll be some curvature, which is another twist on this. But for the moment, let's imagine that it grows in a straight line. Sometimes when it chooses direction and grows, it's going to head and hit a target cell. And that will be a success if it reaches the target cell. But other times it will miss. It will grow in the wrong direction. And exactly the same thing happens in cell mitosis. Microtubules don't know where the kinetochores are. And so experimentally, it's actually observed that they do a search and capture process in which they grow in a certain direction. And then if they haven't found anything after a while, a catastrophe occurs and they shrink back to the spindle, mitotic spindle. Same idea here. In order to deal with failure, we have to allow for the possibility that with some probability, the cytoneme can stop searching and shrink back to the source cell. This is a perfect example of something that's very popular in the theoretical physics community at the moment, stochastic resetting. And so what I'm going to do, I'm going to model this process using stochastic resetting. So this is the basic picture. And you'll notice throughout the talk, I'm using cartoons, but then I put mathematics on the cartoons. So the basic idea is, and let's simplify things, we'll imagine there's a source cell and a single target cell. Sometimes the cytoneme will grow in the correct direction and hit the target cell, but of course it doesn't know where the target cell is. Other times it will grow in the wrong direction and fail. So we have a probability of success and failure, a probability P that it grows in the right direction, and a probability 1 minus P that it grows in the wrong direction. In order to account for this, we introduce the stochasticity to the process that says the cytoneme grows at a constant rate V plus, but then with a certain Poisson statistics, certain probability, at any time, it can suddenly stop growing, shrink back to the target cell, and then begin a new search process in a new direction. And that's the basic set. So there's a growth phase, a retraction phase, and then once it shrinks back, there might be a while it waits before it then starts growing in a new direction. I call that a refractory period. So this is the picture. Let's first of all imagine that I always grow in the right direction. So P equals 1. You'll notice that the equation is quite similar to the molecular motor transport problem, where P plus is the probability density that the cytoneme is growing and here X represents the tip or the length of the cytoneme, and it grows at a rate V plus. But whilst it's growing, there's a rate at which it could switch to the resetting phase. And if it resets the resetting phase, it then quickly shrinks back to the origin at a rate v minus, velocity v minus, and it enters that state at a rate rp plus. When it reaches the refractory state, the source cell, it sticks for a while. And we can model that using another condition, a compartmental equation, which says P naught is the probability it's in this refractory or nucleated state. It enters that state at a rate of V minus times the concentration of probability at the origin. And then it can leave the state and start growing again at a rate minus eta. So that's called a sticky boundary condition, which itself has a lot of interesting features. These sorts of models also apply, for example, to bacteria, where the stickiness is that when a bacteria hits the wall of a container, it tends to stick for a while until it can reverse direction. So a lot of these ideas apply to many different systems. Yeah.
So it effectively becomes a first passage sort of type process.
Exactly. That's right.
Yeah, cool.
And so we have a first passage time problem because
sorry, yeah. I mean, is this the integrated telegrapher's equation with the sticky? Okay. Oh, yeah.
It is essentially that, exactly. Yeah. Is that except I don't allow the minus state to switch back to the plus state. So it's a one-directed version of the telegrapher's equation. Yes. So I can give you a sort of punchline. So now you imagine this process going on. You have multiple targets. To characterize the morphogen gradient, you need to specify the mean first passage times, or the conditional mean first passage times to hit the different targets, and the splitting probabilities. And putting that together is the way you can think about how you set up the morphogen gradient. Okay, so just going through a bit of details. So in order to set up a first passage time problem, you introduce a survival probability, which is the probability that you're still not attached. You haven't found the target. And then using standard stochastic process theory, the first passage time density is just minus the derivative of the survival probability with respect to time, which is the same as the probability flux into the target. Given that, you can then write down the statistics of the first passage time process, including the mean first passage time, which is basically the Laplace transform of the survival probability at S equals 0. Okay, so it turns out, mathematically speaking, even this is an interesting problem to try and work out. And I developed, I think, quite a nice way of thinking about these sorts of problems, which again carries over to lots of other problems, and that is to use basically what's called the strong Markov property. So let me see if I can go through this. How much time do I have left, by the way? So
we ended at 11:30. Good 22 minutes.
What's the time?
Oh, it's 11:08. So you have about
20 minutes.
22
okay,
minutes.
So I won't be able to get through everything, but I like to at least be thorough about what I do get through. So what I'm going to do, I'm going to introduce different stopping times. So curly T is the first passage time I'm interested in. When do I find the target, hit the wall at that distance L? I'm going to call S the first passage time for the first resetting and return to the origin. Now, of course, I might find the target without resetting at all. So in that case, S is infinite. And then R is the time to find the target given that I've reset once. And so there's a picture at the bottom that shows you the idea. This is a sample trajectory. I grow for a while. I then reset. That gives me my S for this particular case. I then wait around for a refractory period, tau. Then I start up again. I could repeat this many times, but irrespective of the second, third, fourth, fifth resetting times, I call the remaining time R. And the crucial idea, which is the use of the strong Markov property, and the strong Markov property is like the Markov property, but rather than conditioning on previous deterministic times, you condition on previous random stopping times, but the same idea applies. And so basically using the strong Markov property, the statistics of R, the remaining time after the first reset, is exactly the same as the statistics of T. And you can exploit that to get an iterative equation for your problem. Okay, sorry. Okay, So I'll now go through this. So how do we exploit this? Well, let me just define some event spaces. Let omega be the set of all events for which the particle is eventually absorbed, which in this case has measure 1. But remember, if p was less than 1, there's a probability that I fail to find the target. So it isn't always measure 1. Let gamma be the subset of events in which I find the target, but I reset at least once. And then, of course, the complement of that is the set of all events where I find the target without any resetting. And the way this method works is that you decompose the mean first passage time into 2 cases. Either this mean first passage time, given that I don't reset at all, and I don't reset at all, or the mean first passage time when I do reset at least once. The first case can be analyzed directly. Because of time constraints, I won't go through all the details. It's in my papers, and I can send someone the talk if they're interested. But basically, I can calculate explicitly the first passage time for the trajectories which never reset, just using the survival property without resetting and the Poisson statistics of resetting events. And so I can calculate that explicitly. And that's just the formula. I just want to give you the basic ideas. In order to calculate the other term, I use the fact that if I do reset once, then t is given by s, the time for the first resetting, the first refractory time plus the remainder. And I can now evaluate each of these terms. And again, I won't go through all the details, but you can use this using the statistics of resetting, the fact that when you reset, you've traveled a certain time at a velocity v plus, and then you have to return at a velocity v minus. That counts as an extra delay, which you can put into the model. That's this 1 plus v plus over v minus term, et cetera, et cetera. So you can calculate everything. But the main point I want to point out is that on the final term of the top equation in the box is the expectation of R. And as I said, from the strong Markov property, that has the same statistics as the expectation of curly T. So the capital T in the boxed equation on the right-hand side is precisely the capital T on the left-hand side. So I've got an implicit equation for the object I want to calculate. So by doing this, I can then use information that I know to calculate all the terms and put it all together and then rearrange, and I get an explicit formula for the mean first passage time of this resetting process that involves the Laplace transform of the survival probability without resetting, which I can calculate explicitly, evaluated at the resetting rate R, together with information about the plus and minus velocities, etc. And it's an explicit expression. So all I need to do is calculate the survival probability without resetting, which I've already done because it's just a ballistic process, and Laplace transform. And so if I do that for this particular problem, I end up with the formula at the bottom of the page. But the point is the penultimate formula in the box applies to many different resetting processes with refractory periods, with finite return times, and so forth. It's a very general formula. So this method of using strong Markov properties and partitioning stopping time into other stopping times is a very powerful method, I think. Okay, so that's some cool math. So I'm not going to do the same thing because it's a lot more complicated, but you can derive the corresponding formula when you allow for the fact that you might fail. And this is where it becomes really interesting because, as I said, the reason that these systems, I think, have a shrinkage phase, have catastrophes in cell mitosis, appear to stochastically reset in the cytoneme context, is that you often grow in the wrong direction and you want to deal with that. And so once you allow for that and include a possibility of failure, what you find is the mean first passage time is minimized at an optimal resetting rate. And this is a general feature of a lot of processes with stochastic resetting. And the concept's quite, and you can prove it directly by using the formula I've written down at the bottom, but the concept is quite straightforward. Without resetting, because I've allowed for failure, there are sample paths, trajectories they never find the target, i.e. they have curly T infinite, which means the mean first passage time is going to be infinite. On the other hand, if resetting is very fast, I never get a chance to travel to L because I keep resetting back to the origin. So again, the mean first passage time is going to be infinite. But for intermediate values of the resetting time, you get finite values, so you have to have at least one minimum. And in practice, for a wide range of stochastic process resetting, you find that this minimum is unique. And so therefore, depending on parameter values, there exists a unique optimal resetting rate that minimizes the first passage time. And this is one of the reasons stochastic process resettings create a lot of interest as an optimization mechanism. Okay. So, final ingredient, we can now combine this with the notion of let's generalize this to the case where we have multiple targets with failure, calculate the probability that when I grow in the direction for the jth target. And now it gets a lot more complicated, so I'm not going to write down any formula, but you can do it analytically. But I just want you to notice the formula for the splitting probabilities. This is the probability that over time, the target I'm absorbed by is the jth target. So go back to that picture where you've got a bunch of target cells distributed in your target domain. Each time you grow in a particular direction, and geometrically speaking, you can work out what's the probability that I'm going to grow in the correct direction. That's the PJ. And then you can work out what the splitting probability is for the different targets. Okay. So any questions about all of that? So you already see a rich mathematical
Can I ask you,
yeah.
So it's amazing what you're doing. In fact, it's it's a perfect thing. I had a student who worked on very similar type of questions, but in discrete time setting.
Yeah.
And he just finished 2 years ago. So I was looking at his thesis here.
Okay.
And this is fascinating. So for example, he was studying the question of doing random search. Okay, from let's say you have a camp and you send out a search party and it either reaches the target and sends a signal, or it never reaches the target. At some point, you decide to send another party.
Right.
Okay. And so if you don't do resetting, then let's say if you're searching on a tree, then there's a possibility that your party will actually wander off to infinity and never get to the.
Exactly. Yeah.
But if you do with some frequency, right? Let's say exponential, whatever, like Poisson. Okay, you send out the another search party, right? Every now and then you make the probability of hitting the target essentially 1. And so the time to the mean time to actually the mean passage time, right, becomes finite all of a sudden. And the other thing is like what you just said about multiple targets, right? If you vary the frequency of the restart, you can alter the target probabilities, and some of them become more probable and some become less probable depending on what the things are, and so on and so forth. So this is fascinating.
Yeah,
thank you.
Exactly. In fact, you can control the shape of the morphogen gradient by changing the resetting rate, for example.
Yep.
Exactly. Okay, one more piece of the puzzle until I'm told to shut up. but I want to get to this at least because I think it's something that I'm amazed that people haven't picked up on more. And at least I want to give you the reason why I think it's important. And that is most stochastic process studies of search processes focus on the search process, the searcher. And there are two types of search. In foraging, the searcher is usually looking for something, looking for a target, maybe a warm cave or food to take. But in cell biology, it's often very different. The searcher isn't looking for something to grab, it's looking for something to deliver. So if you now look at the target perspective, you don't want to just say, okay, the searcher's found me, that's the end of the story. You want to ask what happens over time over multiple search and capture events, because from the target perspective, I want to build up resources and then utilize them. And the perfect mathematical machinery for studying that issue, which at least within cell biology hasn't been studied much, is queuing theory. So I'm probably not going to have time to go through all the mathematical details, but if I at least give you the conceptual framework. So let's take our stochastic resetting search process, but now repeat it. And so we have, if you like, you can think of the customer as the arrival of a cytoneme to a particular target and delivering a morphogen. So the morphogen burst over one search and capture cycle is the arrival of a customer. Then the process repeats. Next time it might deliver to another target if you have multiple targets. But eventually it's going to find the same target again and deliver another morphogen. So you've got a sequence of arrival of customers whose first passage time or first arrival statistics is determined by the search process that I've just talked about. Moreover, these resources are going to be utilized. There's a degradation term. So what one is ultimately interested in, this is, if you like, the way to define the steady state morphogen gradient. You've got balance between the arrival of customers in a queue and then the servicing of these customers, which is an analog of utilization of resources. And this can reach a steady state, which then determines the distribution of resources across a set of target cells. So just as the strong Markov property and the theory of search processes captures what's going on for a single search and capture event, queuing theory seems to be the correct mathematics to study the accumulation of resources from a target perspective. And so then the question is, well, what sort of queue should one use? And the answer is what's called a GM infinity queue. And so when one starts talking about queuing theory, one has to specify a number of things. What is the arrival statistics of customers? What is the stochastic process that services the customers? And how many servers are there? Now, in the case of cytoneme search, the arrival statistics in general is non-Markovian, a general. So that's the G, and so it's a GM infinity Q. That's the G. The degradation or utilization is taken to be Markovian. That's the M in the Q. And because there's no constraint on how many resources are used up, at least in the idealized model, the number of servers is infinite. So there's no logjam. So you can model this distribution of resources across targets through a combination of search and capture and utilization resources using a GM infinity Q. So that's the main. That's the main idea. That just summarizes what I just said. And how much time, Youngmin?
I guess technically 7 minutes, but we did. I realized we did start a few minutes later.
Okay. So let me just sketch the basic idea, because you can use queuing theory now to work out what is the steady state number of resources in a given target. And the way one does it is one basically uses generating functions, which are used to basically determine binomial moments. This is just a more convenient thing to calculate. And so I've defined NT. So this is the NT, the box equation is just a formula for the number of resources in the, I'm just considering a single target here for simplicity, the number of targets in time t, that's given, so given t, only packets that arrived before time t are included in the sum. And then we have to take into account how many of the packets that have arrived have been served. And that determines NT. And the basic idea of how you can calculate things is a bit like what I've talked about, the strong Markov property, is to decompose NT into two terms, one term based on the arrival of the first customer, and then the remainder. And again, we use the fact that the remainder has the same statistics as the original. And so because of time constraints, I don't want to go through all the details, but basically using this decomposition, you can explicitly calculate the generating function for, yeah.
So basically, you're looking at the arrival of the second customer, whether it happens after the first one has been processed or not. Is that right?
No, so in this case, this is an ordered version. So you can modify this where they don't necessarily arrive in order. But this is an ordered case where you are saying, conditioning on the arrival of the first customer, and then given the first customer, I don't care how many more customers there are.
Oh, okay.
Yeah. So of course, the first customer might not arrive, which means n star is also 0. But given that one customer has arrived, it's also possible that several more have arrived. And the several more has the same statistics as the original problem. So you can use that to basically calculate, derive an iterative equation for the binomial moments, which is just an algebraic expression in Laplace space. Again, I just focus on the third bullet point. So I haven't got time for all the details, but I just want to point out that this equation involves this A tilde of S. What is A tilde of S? This is the Laplace transform of the arrival statistics, which going back to the first 2/3 of the talk is precisely the first passage time problem of the search process. So one has a black box, the queue, with these servers. The degradation or utilization is an exponential process, and the input into your GM infinity queue is precisely the first passage time statistics I calculated using search and capture. So now you can see how powerful this is, because you can apply this to a wide range of different problems where you have the same GM infinity queue, but the input, the arrival statistics determined by another complicated stochastic process, a search and capture process. And so you can now combine the two theories and put it together and work out what the statistics are. So let me just summarize the result. Not surprisingly, the steady state statistics is going to be given by the mean rate of arrival divided by the mean rate of utilization. And that's actually a general result called Little's law for queuing theory. And so now you can see that if you go back to the search and capture model, mu is going to be the same, but let's assume it's the same for all the targets. But depending on how far away the targets are, lambda is basically related to the splitting probability of the multi-target search and capture problem. So now you can write down what the morphogen gradient is by taking lambda essentially to be the splitting probability of the search and capture process for the kth target, and then you're dividing by the utilization. But what's beautiful about this as well is you can also calculate higher order statistics, such as the variance and things like that, using this thing. Okay, almost done. I should make one point. Okay, so this is just giving the point that now you combine the two theories by taking the arrival statistics to be generated by the search and capture process. So indeed, this lambda appearing in Little's law is given by the inverse of the first capture time, and then I'm allowing for a dead period before the next search and capture process occurs. And so the rate of arrival for a given target is 1 over the mean first passage time calculated in the first part of the talk plus the dead time, and that gives me lambda. So different targets will have different mean first passage times, and therefore lambda will vary across the system of targets. Okay, and that's basically the idea. Let me just show you a few results, and then I can stop for some questions. So this is how you now put it all together. You imagine a source cell. Again, you can look at lots of source cells and lots of cytonemes, but I just wanted to develop the conceptual framework in the simple case. Let's look at, for example, a one-dimensional array. All one needs to do, besides specifying the stochastic resetting dynamics of the cytoneme, are the probabilities that the cytoneme grows in the correct direction for the kth target. That's a geometrical calculation, depending on the structure, the distribution of the targets. So you can do that using basic trig in the case of a one-dimensional array. So that's then plugged into your theory. And given that, you can now determine the concentration gradient, the morphogen gradient. So let me just summarize this. On the left-hand side of the figure, I show a morphogen gradient for a one-dimensional array of cells with a source cell at a certain fixed distance from perpendicular distance from the array. I have the cytoneme search and capture dynamics I talked about in the first 2/3 of the talk. And then I plug that into Little's law for my queuing model, and I can pull out the morphogen gradient. This morphogen gradient set up just as in diffusion-based models by a combination of a search process, but rather than this sort of diffusion process, it's this search and capture process counteracted by degradation. In the case of diffusion-based morphogenesis, degradation is really the binding and diffusing molecules to the receptors on the target cells, whereas in the case of the cytoneme-based model, it's the actual utilization of resources by a target cell at the same time. And an interesting thing is, as mentioned before, the shape of the morphogen gradient is controlled by the resetting rate. So it gives you another potentially biophysical parameter that could be used to shape morphogen gradients. And moreover, given a particular morphogen gradient, there's another thing one can calculate, and then is, because that's just the relative weighting of the distribution of resources across the array, but one might also be interested in the total amount distributed across the array. So there are two things: how much in total is distributed, and then what's its relative distribution. And what you can show using this analysis is that the total amount distributed is a non-monotonic function of the resetting rate. So you get something analogous to the first passage time. There's an optimal resetting rate at which you maximize the total amount of resources. Okay. One final thing. It's really cool. It turns out a more realistic thing would be to not say, okay, I'm going to just have a single cytoneme growing and shrinking, and I can't go do another search until my cytoneme shrank and started growing again. Much more a realistic model is to say that I periodically grow out cytonemes. I don't wait till one cytoneme returns before I sending out another one. So it's a bit like the camp, people, search parties going out periodically or so forth, which of course is more realistic.
We call it deterministic reset. Effectively.
Exactly. Yes.
You restart at fixed time.
Yeah, exactly.
If you haven't found the target, you go back and start over.
Exactly. And so you can port all this machinery to study this problem as well. Even though there's an ordering that the one that's sent goes out isn't necessarily the order in which searches are started is not preserved by the order in which targets are found, but you can still handle that and do the mathematics. Okay. Whoops. So tell me, should I shut up?
Unfortunately, I think we should conclude.
Okay.
It's not that I want to, but
okay. So I was going to talk about, but it's fine because I've just talked about my own work. I was going to going back to that initial spiel about different approaches to mathematical biology. Actually, inspired by the work I did with Hyung-Jung Kim, a group at Exeter, I think, has developed a much more detailed computational model applied to zebrafish, the neural plate of zebrafish, where it's a lot more complicated because not only do you have all the stuff I've been talking about, but the tissue, the target tissue, is itself growing and undergoing cell sorting. But you can't study that analytically, at least I haven't seen a way yet, but you can certainly computationally simulate it and study, compare diffusion versus cytoneme-based mechanisms. And their computer studies seem to suggest that in certain circumstances, this direct contact process is more effective. And I will end there. So that's just, but just saying that I'm currently thinking about this in terms of cytoneme-based infection. But I'll stop there. Okay.
All right, great. Thank you very much. Let's make our speaker. Are there further questions? I think I think we asked some pretty good questions throughout.
Yeah. May I ask one more?
Yeah, sure.
I just got excited about this. So how do you deal with physical obstacles? I mean, there is a possibility, right, that you start growing and you
Yeah.
hit an obstacle instead of a target.
That's really good. And I've actually looked at that in a different context. And it actually occurs in cell mitosis, because one way to deal with it is to think of another sticky boundary condition. So if you now have a wall, so suppose you have a container, you have the targets you want, and you have a wall. So what can happen, in lines of what you suggested, is the cytoneme grows, but rather than finding a target, it hits the wall, and then it can get stuck at that wall. But then one has to introduce a sticky boundary condition there. But you can then add that to the story of the process. But of course, it's going to make things more complicated. Yeah, so you can deal with that using additional sticky boundary conditions.
Thank you.
Okay.
All right. Okay. Thank you so much again, Paul. This was really great. Let's take your speaker one more time, and we'll conclude there. Appreciate your time.
All right. Unfortunately, I'm not in Florida, so you couldn't take me out for dinner and buy me a very nice margarita, but
I may be in London at some point because our daughter is actually at Imperial.
Oh, is she?
Yeah.
Oh, which subject?
She's a biologist studying biology.
Oh, cool. Yeah,
She's
Imperial is
finishing the first
it's a
year.
great part of London, I have to say. Yeah, so if you're in London, you should definitely look me up.